Well, thank you. Thank you very much for the invitation to the organizers. I appreciate it. That was a fantastic talk by Sarah. I'm going to break some rules because I'm going to talk about some chemistry, but I'm going to hopefully do this in a way that is somewhat interesting. As I was listening to your talk, and I've listened to the previous talks today, it reminded me of something. It's often said that we know more about the dark side of the moon than we do about the bottom of the ocean. In some sense, with ocean acidification, we know globally more about ocean acidification in the open ocean and in most of the world than we do right here on shore. The processes near shore are much more complicated, and we're just beginning to appreciate how acidification may play out.
We have to record you.
Do I need to be up here?
Yep, yes, thank you.
Either one?
Either one.
Okay, sorry. I've got an outside voice. And so because of these complexities, we simply don't understand all of the processes because there are multiple stressors. Now Chris and Philip talked about detection and attribution. I think with respect to open ocean acidification, sort of the classic ocean acidification, we have detection and we have attribution. It's only in the last very few years that we've had any detection of acidification in coastal systems. And it's not necessarily because it wasn't happening, it's because nobody was looking. And so, although ultimately we figured there's a human attribution here, we don't understand the processes yet. So that's sort of what I'm interested in is coastal acidification. So in the open ocean, oftentimes we use buoys like this, sophisticated buoys to measure various parameters. In particular, we can measure the concentrations of CO2 in the water, but also in the air above. And so this is a plot here, and in red is just the atmospheric concentration of CO2. It's right around 380 or 400. So it's pretty constant. That's the current sort of global concentration of CO2. In blue, we see the corresponding surface water concentration of CO2. So they track actually pretty closely. If you look at these deviations of the water from the atmosphere, it's a maximum of about 60 parts per million. So 60 parts per million plus or minus related to 400 parts per million. So I'll come back to that variability. In the Gulf of Maine, we see a similar thing, a similar range. We see that the oceans track the atmosphere, and they track it pretty closely, plus or minus 60 parts per million. So this is a slide that we just saw. I won't spend much time on this, but because Sarah pointed this out, we've got all of these activities that are happening. Many of them are biologically based. Two of the takeaways messages from today's talk I want you to get is there's a difference between open ocean acidification and coastal acidification. The systems are different, although the carbonate chemistry is the same. So that's one point. The other point is that biology really, really matters in these coastal systems to a greater extent than it does in the open ocean. By and large, the open ocean systems are fairly constant. We're seeing this increase in CO2 over time, but coastal systems are much more dynamic. And it's actually the biology of these coastal systems that drives the dynamism that I'll show you here with respect to the acidification, and in particular, the concentrations of CO2 in these waters. And this transition here from blue to green is that I want you all to know we've translocated ourselves from the open ocean to the green productive waters of coastal systems. So this is a map of the Chesapeake Bay. I work for the Smithsonian Environmental Research Center, which is based in Edgewater, Maryland, just south of Annapolis. Here you can see Washington and Baltimore. Down here is the Kegatank farm that Allison will speak about. That's her farm. An interesting thing about the Chesapeake Bay is that it's perhaps one of the most studied estuaries in the world, and we know a tremendous amount about the estuary. However, we know almost nothing about the carbonate chemistry and acidification in the system. So that's the bad part. The good part is because all of those efforts and all the current scientific efforts to understand the bay, it means there are lots of assets and there are lots of opportunities for us to begin measuring some of the parameters that are important for acidification: partial pressure of CO2, pH, the buffering capacity of the water. So they're actually ships of opportunity. There are lots of water quality stations that we could co-locate proper sensors with and probably come up to speed on coastal acidification here quickly. And I would venture to say if we can understand coastal acidification in the Chesapeake Bay, a system as complex as this, we can understand it any place in the world. Yes?
You're talking about coastal waters. Are you including estuaries as
I am.
coastal waters?
I am. So this is something that happened just recently. Maryland, the state of Maryland is concerned about coastal acidification. The legislature's just passed this bill a few days ago, and at this point it's set to create a task force to study the impact of ocean acidification on the state waters of Maryland, including Chesapeake Bay. So there are just a couple features of coastal waters and estuaries that you should know about. They tend to be shallower, lower salinity, but because they're lower salinity, they also have a lower buffering capacity. Now we just heard about riverine inputs can have differential buffering. Maybe some add more buffering capacity to the water. The thing to understand about buffering is that if you've got a highly buffered system versus a low buffered system, a single molecule of CO2 is going to have a bigger effect on the pH in the lower buffered system than the highly buffered system. The carbon flux is much more complicated in coastal systems. It's not just an air-sea interface, but there's a land-sea interface, and there's also an ocean-bay interface, if we think of the ocean course communicating with the Chesapeake Bay. Aragonite undersaturation occurs widely, and this is the issue of waters being corrosive. So we know that aragonite undersaturation occurs widely in the Chesapeake Bay already. Things still live here. We don't understand all of the details. The acidification's effects in coastal systems are not well understood yet, and the biological processes are strongly influencing this carbonate chemistry, which is the underlying chemistry that is acidification. This is a complicated slide, but it's meant to be relating. This is a salt marsh here. This is the waters of the Chesapeake Bay, and this is the ocean. And this is just this is a diagram that sort of describes some of the complexities of carbon flux. And so you don't need to know too much more than there's an air-sea interface, there's a land-bay interface, and there's a bay-ocean interface. And we need to understand how those different fluxes are affecting the system as a whole. This is the Road River. This is a sub-estuary where I work. And I'm going to show you some data here that we've been collecting for the last 2.5 years at the dock, as well as a place here called the Kirkpatrick Marsh. It's also called the Global Change Research Wetland. And in fact, the longest running elevated CO2 experiments on plants have occurred at this very site. They've been going on continually for 30 years. And so the reason I'm interested in this system is that these 2 points are about 1 kilometer apart. But as you'll see, the dynamics in terms of the ocean acidification dynamics, or in this case, the concentrations of CO2, are very different, even at locations this close. So this is, again, it's a sub-estuary. It's got an annual salinity range of about 3 parts per thousand to 15 parts per thousand, and that's related to, say, 35 parts per thousand, which is fully marine water that you'd find in the ocean. This is a system that we use to actually measure the concentration of the water itself. And essentially what we're doing is we're pumping water through this apparatus, and we're bringing the headspace in these tubes here into equilibrium with the gas in the water. So we're basically, essentially, you can think of, we're shaking the gas out of the water, and then we're measuring an air sample. And that's got a real advantage in that our sensor's not dipped in the Chesapeake Bay, which during the summer, if anybody's had a boat, is fouled with all sorts of organisms and algae very quickly. Biofouling is a real problem. And although we've got extensive pH measurements in the Chesapeake Bay, we don't really know what those mean, because there are a couple of problems with measuring pH. One is this fouling. The other is we're measuring with probes in a system that's changing salinity all the time. And pH probes don't like that. So what we did this is we did this both ways. This is actually a pH probe right here. So we run the water by the probe, and then we run it through the system, and we measure the CO2. This is about a 2-month plot here. And so in green is actually the pH measured from the probe. In blue is the corresponding measurements of what I call PCO2. It's the concentration of CO2 in the water. The red line here corresponds to the atmospheric concentration of CO2 around 400. And if you recall, I said in the open ocean, it's 400 plus or minus 60 parts per million. Well, we far exceed that range on a daily basis. These black bars are night. These white bars are day. And I'll show you in a couple of slides why that's important. What we see is we see these stereotypical changes in CO2 concentration. During the day, photosynthesis draws down CO2. And at night, when the lights go out, photosynthesis stops, but the organisms continue to respire CO2. So it rebounds. And so you can see from here that even across this period of time, we've exceeded the range that you see CO2 changing in the open ocean by a huge amount. We see the changes that are much bigger in a day than we'll see in the open ocean over 2 years. The red bar is what I just showed you, early sort of January. This is out to July. And so what happens is and this blue bar here, that's atmospheric plus or minus 60. So that's what's happening in the open ocean. This is all to tell you these coastal systems are very different. The dynamics are quite different. So here we're peaking out at not 400 parts per thousand, but 4,000 parts per thousand, or per million CO2. So that's an order of magnitude, 10 times the quantity of CO2, the concentration of CO2 in these coastal waters. This is last September, again, night and day, and it's all to show you, this is the tidal height. So the tide goes up and down, day and night. This is, when we measure at the Cirque dock, we don't see any tidal signal. What we see is this photosynthetic and respiration signal. Photosynthesis pulls down CO2, respiration brings it back up, and that happens consistently on a daily basis. Well, we've also measured CO2 at the salt marsh nearby, and what we see there is we see a different sort of pattern. It's simultaneous measurements in locations that are within a kilometer of one another. And if you actually looked into this, you'd see that this tracks the tidal cycle very closely, and you don't see a day-night cycle. So the other thing is that the concentrations are much higher. This is a scale of 20,000 or 25,000 versus 2,000. So what's happening? Well, it's not just an air-sea interface. What's happening is waters at high tide goes and inundates that marsh. They're high centers of productivity, but they're also high zones of decomposition. So that decomposition, all the CO2 from that decomposition goes into the water and gets washed out into the river. So we see these very, very high CO2 signals at low tides and signals that represent the river at high tides. So it's a very different system, but within a kilometer. This is just to show you in purple, this is what's happening at the marsh, and that's the signal at the dock, a kilometer away. Someplace buried in there is the very constant signal in the open ocean. So coastal systems are different than open ocean systems. This is just what I told you about outwelling of CO2 can be driven tidally. But it should tell you that we've got to start considering more mechanisms. We've got to consider tide, we've got to consider photosynthesis, we've got to consider respiration, biological drivers. This is just sort of the corresponding pHs that we see at the dock during that same month. These are individual measurements that we took where we did wet chemistry and did careful calculations of the pH. All a bit less than 8, but greater than 7.5. These are all at the dock, and this is just the up and down of the tide. So the tide doesn't have much effect on this pH. However, if you can see these mustard-colored dots, this is the corresponding pH that occurs at that marsh in the tidal creek. And so what's happening there is these high spikes in CO2 result in very low pH values. So we see the pH swinging around much more wildly. And again, this is within a kilometer. In the open ocean, you can have a buoy that's representative of hundreds to thousands of square kilometers around that buoy. If we stick a buoy in the Chesapeake Bay, it's not going to tell us much. We can see what's happening. So we need more measurements in the bay. We can't use expensive buoys. We need to come up with systems that are cheap, that are good enough, and that give us the information. This is just some of those points I was just making. Just to move on quickly, I want to show you a couple of results from some experiments we did with both larval oysters and juvenile oysters. And this is the eastern oyster, the native in the Chesapeake Bay. And these experiments were done in an estuarine setting. Most studies are done at fully marine levels. And also, this is a closely related species to the Pacific oyster, Chrysostria gigas, which we've heard about in the Pacific Northwest, which went through a severe crash in hatcheries. So I'll concentrate on the larval stage in the first set of slides, and in the juvenile stage, the post-larval stage, in the second set of slides. The first set of slides are laboratory experiments. The second set of slides are from field experiments. So these are just the experimental conditions in the laboratory. The most important thing here is that we had different CO2 treatments. So we had a 380 part per million, which represented sort of current atmospheric conditions. We had a 50-year projection, which is a 560, so it's a more enriched CO2 system, and an 800 part per million. It's called microatmospheres, but it's similar. You can think of it as parts per million. We also induced a pre-industrial concentration, because we could do that in a laboratory, where we imposed 280 parts per million, which is sort of reflective of what happened before we started burning fossil fuels. And the results were interesting in that what we saw was that in the pre-industrial and in sort of the current levels of CO2, we found that the oysters grew faster and they calcified more than the ones that were grown at high CO2 levels. And so, interestingly, this is not necessarily the case across all oysters, or even all oysters that are of this genus Chrysostria. Gigas, I think, is more sensitive to CO2 than is Chrysostria virginica. So that's another complication, is that there are going to potentially be species-specific effects of acidification. And that's probably largely driven by the ecological setting and the evolutionary history of where these individual organisms evolved. So one of the things that we noticed when we looked at these larval oysters under an electron microscope was that they had these growth rings. And so when we compared the number of growth rings on a high CO2 versus a low CO2 oyster, we found that the same number of growth rings. It turns out they're daily growth rings, very similar to the growth rings in a tree. And so we did an analysis of those. We counted them, and then we counted the spaces in between those rings. And what we found was that the oysters that were grown under low CO2, the pre-industrial setting, versus the end of century projection, there was much more growth between each ring. They were growing faster. And so despite that, we didn't see any sort of severe deformities in these oysters. So it's not like oysters and biological organisms are inert. They can do homeostasis, all this sort of thing. So they have some control of their growth and their environment to some degree. And so it's important to take that into consideration. But I think what's happening here is that it's an energetics. The ones grown under high CO2 have to use a lot more energy to make that shell, in which case they're growing slower. And ecologically, if you're growing slower, it means that you're in the water column longer, and it means that your ability to actually settle and become an adult and add to the next generation may be diminished. So there can be real ecological repercussions here for slower growth. This is the final couple of slides. This is where we took baby spat oysters and we superglued them to these plates, and we put them out in various places in this river that I showed you. At the dock, at the dock, at the marsh, and then someplace in between. And so what we found was at the dock, we planted them in September, so it wasn't the best time to do it, but we let them overwinter, and this is what they looked like at the dock after 8 months. This is what they looked like in the creek after 8 months. They were still alive, barely, but they weren't growing or calcifying at all. And these were the ones in the middle, sort of intermediate growth. Also, mortality was much greater in that tidal creek. Now, it's not to say that oysters can't grow near salt marshes, but if we think from a restoration point of view, we spent a lot of energy trying to restore oyster reefs in Chesapeake Bay. I don't think there's been any consideration of the carbonate chemistry when we think about how we cite those things. If it were up to me, I probably wouldn't try to grow an oyster reef in the Kirkpatrick Marsh tidal creek. The chemistry's not in its favor. So I think we need to begin to take into consideration some of these issues of acidification when we think about restoration. And this is the final slide. So just in terms of coastal acidification and context and some considerations, when we think of habitat restoration, I think it's going to be important for oyster reefs, but it also turns out that acidification affects seagrass growth and the chemical composition of seagrasses. Seagrasses is another really important habitat in the Chesapeake Bay, which, by the way, are important carbon sequestration organisms. Also, what's happening in tidal marshes, restoration of tidal marshes. These things are all linked in a place like the Chesapeake Bay, and we need to take that into consideration. So carbon capture and sequestration, shell fisheries is obviously an important thing that we need to think about. Additionally, fisheries in general and aquaculture. As the population increases and we enter sort of into this higher and higher CO2 world, we've got to come up with a way to feed everybody. We're not going to be looking inland at livestock. We're going to have to turn around and look to the oceans. And so it's going to be important that we understand this chemistry. Many of these fisheries rely heavily on these coastal systems. So I submit that it's important to understand how acidification is playing out in these systems because they're going to be long-term repercussions. And finally, because I'm an invasive species biologist, I have to say that the natural assemblies of organisms in a place like the Chesapeake Bay or any coastal system are going to be affected by acidification. If we begin selecting against calcifying organisms, things with calcium carbonate, body parts, shells, corals, what's going to happen is we're going to change the nature of those communities. Some of those things are going to go extinct, and it's going to open the door for biological invaders, things that were today may be repelled by biological pushback of those natural systems. If those systems are compromised, we're going to see increased invasions in the future. So with that, I'm sure I've gone long. Apologize. Thank you.